Welcome back to What Did I Do in Lab Today, where I tell everyone what I did in my entomology lab today. So last week I collected these specimens to use in the Wolbachia assay, where we're basically just screening for the presence of Wolbachia bacteria. So I collected those were sitting in ethanol for basically just a week to preserve them while we just waited for our next lab section. So today was the second part of that, where we were doing basically DNA extraction and preparation for the rest of the lab. So what we did, we had those 3 samples, like each person had 3 samples, so me and my lab partner had a total of 6 samples. But you need just the DNA from this, so we wanted to get that out and leave the rest of the bug. We don't want that, that's junk. So kind of what we did, it was a decently long, well, not super long process, but it was like a couple hours that we had to be there. And so what we did, we had the insect samples, we put them in those little colorful tubes up there, and we had to masticate them, which basically smush them down to release all the cells and put them into a solution that allowed the DNA to kind of be extracted into it. Just like release all the DNA from the cells in that. So we had to put a bunch of different chemicals in, mix it up for a pretty long time, do a bunch of steps. Then we had to leave them to incubate for 45 minutes at 55 degrees, and then we had to add some more chemicals and incubate them for longer at 75 degrees. So at this point the DNA should be good and ready to extract. So we added another chemical and then centrifuged it this time, which is different from the vortex that I just showed. It's a machine that basically just spins things really fast to get all the solid stuff stuck to the bottom, or in this case in a elution tube. Basically that sent all the liquid to the bottom while catching the solid stuff in the tube and then just separating it that way. So basically what the purpose of this lab was to get the DNA out of the bugs. So basically we just broke open the bugs, broke open the cells, and extracted the DNA, which is now ready to be stored, and we will use it next lab to amplify it, which basically, oh my god, I can't talk, which basically means just get more copies of the DNA through PCR, which I might have explained here. I think I talked about it briefly multiple times, but yeah, we're gonna do PCR next week to get more DNA to finally do the Wolbachia screening. That was a struggle to get out, my god. So basically, what we started with was a bunch of bugs in tubes, and now what we're left with is a bunch of bug DNA in tubes.